Morning. Good morning. My name is Gus Kasulis. I'm the principal investigator of the Louisiana Biomedical Research Network. This is our third day of the virtual conference in bioinformatics and computational biology. It's the first time we do this virtual conference for those that may have joined us just today. We're spread over 3 days. Typically, we have it for a day and a half, and hopefully that will happen next year after we're done with the pandemic. We have been delighted to listen to expert seminars from all over the country and having attendance actually from a number of different states, as well as a few from international places. So without further ado, I will leave it to the core, Dr. Ramesh Subramanian, our coordinator, and Chris Taylor to introduce the speakers and move forward for the third day.
Right. Thank you, Dr. Kasulis. We've had pretty impressive past 2 days, and we are very excited that our program today kicks off with Dr. Moriah Szpara, who's an associate professor in the Department of Biology and Biochemistry and Molecular Biology and the Huck Institutes for Life Sciences at Penn State. She did her BS from Penn State in biology and then went to UC Berkeley, got her PhD, and then worked with Dr. Lynn Enquist in the area of neurotrophic herpes viruses. And then came back to Penn State again. So back where she started her undergrad career, so in biology. And Dr. Szpara has been working on genetic variation and adaptability in HSV, and also the neuronal consequences of infection, potentially leading to Alzheimer's in the later stages. And recently, she's also been doing a lot of town halls in response to the COVID pandemic, trying to spread vaccine awareness. So she's got her fingers in a lot of pies, and it's my great pleasure to introduce Dr. Moriah Szpara. Moriah, it's all yours.
All right. Well, thanks, Ramesh and Chris and Gus and all for the invitation to come and talk with you here today. Let me start up my screen share. So this has been a really fun conference. It's not one that we have going on here in Pennsylvania, but I think it would be a great resource to be able to draw in more trainees and give them a broad exposure to a whole range of different bioinformatics and genomics areas of application. So I'm going to tell you about some bioinformatics and genomics applications in the world of herpes virology. And I like to start out by thanking all of the folks who actually did this work. So that's the lab, as you see it there, plus a couple of recent graduates who I've sort of put onto the picture, and of course our funding agencies. And okay, I'm going to start off by just giving you kind of a quick broad picture overview to say that we're all living in a viral pandemic, right? So whether or not you studied viruses before, you probably have an appreciation now of how much they shape our world generally and then shape our lives personally. So acute viruses are more often RNA viruses and ones that have an infection that might last for days or weeks. And for most of us who would have been exposed to SARS coronavirus 2, it is that sort of short-term infection. There are some cases where it becomes a long-lasting infection, but down here below, I've just put the sort of global spread of this as of February. If I put the March statistics, it really wouldn't look much better. But I'm going to talk to you today about a different group of viruses, ones that are perhaps not making headlines, but are ever present and always with us. And those are the chronic viruses. So more often, these are DNA viruses, not always. As I mentioned, SARS coronavirus can have long-lasting effects. But these viruses on the whole will last weeks, months, or even a lifetime. And the ones I'm going to talk to you about today are the herpes viruses here. And actually, I'm going to pause for a second and switch to the laser pointer to make that a little easier. So the herpes viruses are the ones that I'll talk to you about today. And just to give you an impression of the scope, I made a small map for you down here to show you the overall seroprevalence just for HSV1, which is just one of those 9 species of human herpes viruses. And you can see that it's just as widespread. The only places in gray on the map are the ones for which we just don't have statistics. It's not that there are actually no herpes viruses or no HSV1 there. So among the chronic infections, I wanted to give you an overview of the different types of those that would infect humans. And there are chronic viruses for all animals that we have looked at. So those chronic viruses that any of us might carry or do carry would exist in any of a number of different niches. So the oral niche, the respiratory niche, the GI area. And among those different areas, there will be multiple viruses coexisting. And I've highlighted for you here in red where the herpes viruses would be, but I'll dim out the others and make that focus even more broad to say that the two viruses I'm going to talk about the most today are two of the human herpes virus species, HSV1 and HSV2, so herpes simplex virus 1 and herpes simplex virus 2. And those are most commonly found in the oral and genital niche. They can also occur on the skin in the format of what's called herpes gladiatorum or herpes whitlow. And I should mention, actually, I'll go back a second to just mention that HSV1 wasn't as commonly found in the genital niche in all countries around the world. Here in the United States, this is a relatively new epidemiologic trend to see HSV1 primary infection occurring in the genital region as well as in the oral region. And there's a number of public health trends that underlie that. But the observation is that it's a new niche in which we need to consider this pathogen now co-occurring with HSV2, which was always found in that niche. Albeit in different individuals. So I'm just going to pop up my screen here at the top. It's making it a little bit hard to see. What I wanted to do first here was to give you an overview of the different human herpes virus species. So there are 9 total, and I've highlighted them for you here in red, but also just sort of blown up on this phylogenetic tree, the alpha herpes virus groups. And the alpha herpes virus groups include, viruses that infect pretty much every species you can think of, both mammalian and bird. But the ones that infect humans are HSV1 and 2, which I've already mentioned, and varicella zoster virus, which is the causative agent of chickenpox in children or shingles. And many young people today who are now in the 20 to 30 age bracket were vaccinated as children, but I'm old enough to have had natural chickenpox. And so there's those of us walking around the planet who've had natural infections, as well as those who are vaccinated and may need nothing more than a booster later in life to totally prevent this infection lifelong. But one of the big distinctions between the alpha herpes virus part of the tree here and the beta and gamma herpes virus part of the tree on the bottom is where they spend their lifelong latency. And I'll illustrate this for you on another slide in a second. But the alpha herpes viruses have a lifelong latency in the nervous system, whereas the others, the beta and gammas, have a lifelong latency in hematopoietic stem cells that are in the lineage that eventually would form B cells. So they're circulating in the blood and moving through the bone marrow, whereas the reservoir for lifelong latency for alpha herpes viruses is not accessible in humans. We don't cut neurons out of living humans, and so we can really only sample these viruses in a more limited way. And I'll tell you about some of those approaches today. But first, just to give you an overview of what a natural infection would look like, at least in a diagrammatic fashion. This is a depiction of a human with an HSV1 or HSV2, it could be virion, at an oral niche. And so the virus is going to initiate its infection in that oral niche, or remember I said it could be genital or really anywhere on the skin. But what happens then is that there's a localized infection, let's say in the oral mucosa, and then the virus is actually taken up by sensory or sympathetic nerve endings that are in that oral mucosa and are innervating that region. And when the virus reaches neurons, it has the ability to set up a different kind of infection that we call latency. And during this infection, it's worth saying that while these viruses are lifelong, they're different from human immunodeficiency virus or what you might have heard as an endpoint, let's say, for human papillomavirus, in that they're non-integrating. So they will stay as episomes, so free circular DNA inside the nucleus of those neurons, but they're not going to integrate and damage the host genome. And most of the time during latency, there's no overt symptoms to the person who's carrying that virus. But what you do have is this lifelong neuronal reservoir, and the immune system, while it may provide surveillance of those infections, won't clear or kill those neurons. And so you can have times of stress where the virus basically reactivates, and a number of virions are made inside of those neurons, which then transit back to the oral mucosa or whatever was the initial site of infection, form a lesion, replicate additional rounds, and then potentially spread to new hosts. And so those are the recurrent painful lesions that people who are suffering from outbreaks of HSV1 or HSV2 will experience. But I hope you can see that there's a whole lifetime that the virus is spending in the body that is not so noticeable to the humans that are carrying them. And just to give you a sense of the public health scale of these infections, HSV-1 infects billions of people around the planet. The seroprevalence is over 60% in most countries and up to 90% in some by adulthood. HSV-2 is far less prevalent, but we're still talking hundreds of millions of people. So these pathogens have been co-evolving with humans for a long time, and they're not going to go away anytime soon. So they're certainly not making the headlines that SARS coronavirus is right now. And I'm immensely grateful that such a prevalent virus is not usually lethal, but that doesn't mean that it's pleasant for the people who are walking around with it. And as I mentioned before, HSV-1 is causing more new genital infections right now than HSV-2. And that's due to rising prevalence of oral genital interaction. So the rising prevalence of oral sex is causing more exposures of HSV-1 in the genital niche. But we're concerned that this might lead to adaptation and changes in these pathogens that would not be a good thing from a human health perspective in the long run. One of the things that's interesting about this pathogen, but many people have become more familiar with this year in the context of SARS coronavirus, is that not every person who gets infected with HSV-1 or HSV-2 will experience the same symptoms. So some people will experience a more frequent rate of oral or genital lesions. You can also have the virus occur in different niches in the body, as I mentioned before. I didn't actually mention that this virus can be introduced into the eye, and it could cause ocular scarring and vision loss. And so that is, at times a self-introduction from the oral niche to a person's own ocular site. But in any case, losing vision due to an infection like this is an extremely serious morbidity for this pathogen. And in neonates, as I mentioned here, that is actually a population where this infection can be fatal if it's not caught. And so there are 3,000 or more infections per year in the United States. And the ones that are not caught don't end well. We've published other work on that aspect of infection, but I'm not going to have time to talk about it today. So feel free to ask me about it at the end if you're curious. And I'll just add that right now we have a lot of candidates in the trial pipeline for good vaccines that would be preventative or therapeutic, but nothing has succeeded thus far with any significant efficacy. And I should just add for the young folks who might be listening that, in the age of many of our trainees and the ones that I have talked with during the course of this conference, that half of the new sexually transmitted infections in the United States are happening in young people. So I might have built a research lab and be studying this area, as is Gus and many others there. But the most impacted population right now is actually young people. And because these viruses are lifelong, those young people are going to be dealing with those infections for the rest of their lives. There are other sexually transmitted infections that are bacterial and are curable. This one's not. And we actually don't have as many drug opportunities for this infection, or available drugs, I should say, as we do for HIV. So there's a real critical need to provide better ways to control these infections for the many young people who need to manage them for many years to come. So I want to give you just a little overview about the virus itself before I take you through some of the exciting new data that we have. And I always like to start with a picture of the virion because it's kind of like the way a human looks in the mirror. It's good to look at the thing that you're going to study. So on the right are some actual images. These are electron micrographs of actual virions. And on the left is a perfectly spherical, perfectly symmetrical textbook version of a virion. And one important point that I'll say here and mention again later is that these are DNA viruses. So I said that earlier, but I just want to emphasize that. So they're double-stranded DNA, and they pack that DNA in an icosahedral capsid, but then bring along a whole tegument layer containing a whole bomb's worth of viral proteins that help to take over the cells that the virus enters, and this surface that is studded with viral glycoproteins. And that, you might think, looks a lot like a SARS coronavirus virion, which also has an even more glorious coat of enormous protruding surface proteins. The herpes virus ones don't protrude as far, but are just as actually more numerous and more complex and just as important in the virus's biology. But I include the ones on the right in part to point out that just like humans, viruses are in real life lumpy and asymmetrical, and each one slightly different from another. And you'll hear that repeated as I go through the talk, because these viruses, it turns out, are really as different from one another as each one of us is from each other. We all have different hair color, we all have slightly different appearances. And so it turns out do these extremely common human pathogens. So this is where I'll use this slide as a place to come back to as we take you through some of the data from the lab to give you kind of a flavor of the different things that we have seen as we look at these viruses and look at them closely and try to notice their individuality. And then the ways that genomics has really informed all of this, because it's been a great way to crack the lid off of what we can see with our eyes or see with a microscope and see the genetic differences that underlie the things that we can see with our eyes or with a microscope. So I'll start off with just looking at the viruses and showing you what it looks like when you examine them and look for the fact that they're not all perfectly alike. So like most virologists, we have to bring viruses into the lab in most cases to study them. And the history of virology is built on the ability of scientists to isolate pathogens and bring them into the lab and study them so that they could make sure that the virus they were studying was the thing that caused the disease that they were trying to study or prevent. For those of you who have not seen this in a virology talk before, or had the opportunity to work with viruses in the lab, I wanted to show you this extremely common technique that virologists use, where in order to study viruses, we often take those viruses and put them in a dilute solution, and then put them on a monolayer of cells that the viruses are capable of infecting. And so what you're seeing here is actually a single focus or plaque, and I'll show you these in a live movie in a second. But what I'm showing you here down below is a much more zoomed out image. It's actually a microscope image of 7 by 7 images from the same microscope that have been stitched together and overlaid seamlessly. And that's to show you more like 40 or 50 of the same sort of viral plaques that are in the upper right of the screen. And what I just want to point out is that when we normally work with them, we actually stain them in a way that looks like the image on the left, where after we've allowed those viruses to land on the susceptible cells and form plaques, we stain the cells with a dark dye called methylene blue. But that dark dye and these white spots are a little bit hard to see on a screen, so I actually inverted the images so that you're seeing inverted images here and in the other slides in the presentation. So I just wanted to make that clear for those of you who are virologists and might wonder why it looks backwards. These images on the right are a little easier to see. But I promised I'd show you what it looked like live. So this is now on the left, the movie that's playing, a single plaque that's forming in a monolayer of these susceptible cells. And the one on the right is also a plaque forming on a monolayer of susceptible cells. But I hope you can see that those two things looked really different. They're actually from the same source population. It's from a strain called KOS or KOS. And I'm going to restart these movies and I'll play them at about the same time. Okay. Somehow I have lost my ability to play the movies. Okay, there we go. So now they're playing at about the same time. And I hope you can see that the one on the right, which is doing what's called syncytial spread, actually spread faster than the one on the left. So the one on the left is actually a more standard way of a virus spreading. It starts in an individual cell, and I should say this movie started 20 hours after infection because I had to be able to find where the virus had landed on the dish. But it starts in an individual cell, makes many copies of itself, and moves to the neighboring cells, and so on. And both of these plaques are existing in this monolayer of infected cells under a viscous substance called methylcellulose that makes the virus have to spread sideways as opposed to diffusing through the fluid layer and moving anywhere in the dish. But what happens is that when you have a syncytial plaque, the virus isn't going from one cell to another in a way that involves killing the first cell. It actually just fuses with the next one, which in turn fuses with the next one, and so on until you have this enormous multinucleated cell, which at some point will actually pop. And that's where you get that interesting phenomenon where right about here, you'll see the membrane is so tightly pulled that it just snaps and moves outward. And so both of those, I told you, came from the same initial mixture. It's this example here called KOS. But when we started looking at plaques and looking at them carefully, we realized that almost every virus strain we looked at had a mixture of plaques. And most virologists would tell you, well, sure, they're there, but it's probably just where the plaque landed on the dish. It's not a big deal. Just pay attention to the ones that are the majority. But I was a curious postdoc, and so I wanted to know if I could separate them out. Was it really just where the plaque landed in the dish that made this one bigger or look syncytial? That didn't quite stick in my mind. And it turned out that if we picked one that was an ordinary size and grew it alone, it would make more plaques of an ordinary size. And if we picked the ones that were large and syncytial, like this one on the left, and grew it alone, it would only make things that were large and syncytial. And so that was really an important way to demonstrate that there must be something genetically heritable about these differences. It wasn't just that you took a bunch of identical viruses and threw them down in places that were more or less susceptible. They really carry genetic differences that could be propagated. And I'll come back and tell you about the genetics underneath that in a second. But one of the next things that we did more recently was to ask, okay, if this is genetically heritable and you can have a virus population from one person that has all these variants, what happens if you let them compete with one another? So we set up a situation where we did what's now called experimental evolution, kind of watching evolution happen in a lab setting. So we took that mixed group of viruses and we actually bottlenecked them a little bit. So we took a population of mixed viruses and then diluted it to a very, very small dilution and put it on another monolayer of susceptible cells and repeated that process 10 times over. And each time we watched what the final titer was, counted how the plaques looked and if they were syncytial or not, and sequenced them. And interestingly, what we saw was that those large syncytial plaques that were kind of rare at the beginning, like this one, they took over. And by the 10th passage, they were nearly 100% of what was present. And so what I'm graphing for you here in these colored lines are 3 lineages where we did this competition, and each time syncytial viruses seemed to win or to take over. And I'll tell you about the genetics that underlie that in a second, but I just want to take a pause there in case there are any questions that people want to ask in the middle, to say that that was really our first set of lab-based studies that showed us that there are definitely differences hard-coded into these viruses that can even start from the same initial stock from the same infected individual. And that tells you 2 things: that infected individuals have a mixed population and that population is undergoing competition and selection, and that we can study it in the lab. So if anybody has any questions, feel free to ping in or raise your hand, or John can jump in and tell me, because I can't actually see the full list of participants. But otherwise, I'll go on to tell you about what we saw when we deep sequenced those viruses. So to talk about the deep sequencing data, it's worth just pausing for a second before I dive into the data to tell you about a sort of paradigm in the world of virology. And that paradigm is that DNA viruses are really stable and don't usually have the range of genetic diversity within a single species that I just showed you. So this is a really classic plot from a particular paper I've quoted down here, but it's been replotted many times and repeated many different ways. And what this plot is showing you is that as genome size increases, in general, the observed mutation rate at the level of single nucleotide replacements or differences between individuals goes down. So we have a relatively low mutation rate as compared to, let's say, RNA viruses. And many RNA viruses like poliovirus, rotavirus, many others, do not have any proofreading capability in their genomes. SARS coronavirus and this pandemic that we're living with is notable because it's one of the only RNA viruses that does have proofreading. And so we as humans are ridiculously lucky. Well, not lucky that we're having a pandemic, but at least lucky that the virus has not been mutating even faster, because that would have really made the outcomes even worse.
So
but
I
for my
don't see
purposes,
any questions, by the way, in
okay.
chat or anywhere, but if someone puts one in there, I'll try to let you know.
Okay, perfect. Yeah, because I'm happy to do questions in the middle if people have them. So the reason I wanted to put this out here today was partly to remind you of the context in which we're working, but also to say that I think the data I just showed you demonstrates that DNA viruses do have the ability to be quite diverse. And in part, it's because these kind of plots about the mutation rate for both higher eukaryotes like our cells and bacteria and viruses overlook the fact that there's lots of other ways that diversity comes into populations. It's not just about how often your polymerase makes an error or fails to correct an error. Recombination is another huge source of diversity. So are fluctuations in tandem repeat lengths. And the competition and selection I mentioned also matters. So many of you might be studying other pathogens and other model organisms, but it's a good reminder for all of us that when we oversimplify to only talk about polymerase error and single nucleotide replacements, we're leaving out a lot of biology. So the reason that I mentioned those tandem repeats there is not just because it's a true statement that they drive diversity, but also because these viral genomes contain hundreds of tandem repeats. They have more tandem repeats per kilobase than the human genome, even though our genomes, relatively speaking, compared to other mammals you might think of or avian species, we still have a lot of repeats. These viruses have even more. So this is a depiction of the viral genome here, where there are large swaths like these, UL and US stand for unique long and unique short, that are flanked by large repeats, these kind of blue regions, but also past all of the coding regions that are depicted here, they have these hundreds of homopolymer repeats or microsatellites and mini satellites, which are larger tandem repeats, scattered throughout the genome. And this entire virus is highly GC biased, but that bias is extremely high inside of these repetitive elements. And I mention that just to say that sometimes we work with genomes that are extremely challenging, and this is certainly one of them. Other herpes viruses are actually easier to sequence and therefore have had more genomic progress made in the last few years than HSV. So when we started working with these genomes, we realized that a lot of the tools that had been built, let's say for the human genome or for other model microorganisms like yeast or E. coli, weren't working well for these viruses, in part because they were GC rich and because of all those repetitive elements. A lot of the existing tools would break or work suboptimally and really only give us fragments of the viral genome. So this is an overview from one of our papers. It's in the supplement. So in case you like the figure or want to remake something like it for your own purposes, it's not in the main paper. You have to actually pull out the supplement to find it. But it kind of takes you through what genome sequencing really is, which is, starting with some sort of piece of DNA or RNA if it's a coronavirus, quite often shearing it up in order to make it work with a lot of the short read sequencing technologies like the Illumina approach that we use. And then working with assemblers like Sake and Celera. Those are the two that we've used most often to kind of put those contigs back, put those sequences back together into contigs, and then fill in the gaps and come up with a draft genome. And I'll tell you what we've seen as we did this for many, many different approaches, but I just wanted to say that we have over the years built both command line tools that we commonly use because they're very customizable as we push the envelope for what's possible, and also web-based tools like this one called VIRAMP, which exists in a Galaxy environment that I think has been mentioned here. VIRAMP is actually running on Amazon Web Services right now. And I would say more people use it for RNA viruses than we do in the herpes virus world. So we built it for HSV, but a lot of people have found it to be useful for other purposes. And I think that's a common theme when it comes to bioinformatics is that if you address one need, you might realize it can fill a bunch of other purposes and it will evolve and grow over time as well, and go far beyond what you envisioned. So I'm going to tell you some of what we saw in those genomic studies. And for that, I just want to remind you of these sibling isolates that I had told you about earlier, where I said we took a mixed population and we proved that we could pick individuals and that they would kind of replicate with the same appearance that they had at the start. And there were two strains that I showed you, COS and F. They were actually in the opposite orientation of vertical versus top and bottom on the other slide. But I'm going to zoom them out here in order to put up a phylogenetic tree from one of our earliest rounds of sequencing a number of different viral populations. And it was in this paper that we had sequenced these groups of what I call siblings and wanted to see how related they were, how many genetic changes actually existed at the DNA level. And what we discovered was that sibling viruses were actually pretty closely related to one another. On average, they were about 98% identical. And I have to move my Zoom window here on the side to get us to the next part. But what was interesting to see was that when we looked at, let's say, the group of F siblings compared to the COS siblings, they had a lot more genetic difference between them, on the order of 3 to 4% of their genome. And if you think about that in terms of the actual proteins, these viruses have really tightly packed coding regions. So they don't have a lot of space where they can have genetic differences between isolates that aren't impacting coding sequences. And as this group in particular knows, even changes in non-coding regions can have regulatory effects on gene expression or timing thereof. So just to put this in context, humans and chimpanzees also differ in about 3 to 4% of our genomes, particularly when you take into account these tandem repeats in addition to single nucleotide differences. So what we're really seeing is that these different viral populations that come from different human individuals potentially have as much phenotypic difference in space between them as Jane Goodall and her favorite study subjects. And I think that that's just a good illustration to kind of take home as the first pass. What did you see when you actually sequenced those individual clones? And then I'm going to dive in a little bit deeper. So this is the next pause in case you want to pop a question in there and John can interrupt me. But otherwise, I'm going to dig a little deeper on what else we saw besides that kind of surface level difference between lineages. So you might have noticed, if you're a person who actually works with genomic data, which I think a good number of the individuals listening today are, that I had a diagram like this when I was talking about the genome sequencing kind of flow chart. And what this is a depiction of a consensus genome, a black line, with kind of a cartoonized coverage plot up here for how much depth of sequence coverage we might have. And this is a cartoon of individual sequencing reads aligned to that consensus genome. And what I'm illustrating for you here with this little arrow is a place where not all the sequencing reads agree with what the base should be at that particular locus. And this is more of a real life illustration from an actual IGV or IGV genome browser, where pink and blue are reads facing in opposite directions. But what I just want you to see in this squashed view is this is a particular place where there's support for 2 alternative nucleotides in that position. And when we build a consensus genome and we say, this is the average genome, it's on average a G in this position. I'm not showing you or anyone else the fact that there was actually an alternative population in there that had perhaps an A or a T in that position. And to show you why this matters, I'll use this very cartoonized diagram on the right to say that if these are individual genomes where A's and T's and G's and C's are kind of turned into stars, the consensus genome on top is an accurate representation of the population. But I'm going to change around these lines without changing the underlying data. And I hope you can see now that even though the consensus genome is a fair representation of frequency, it is not the best representation of the most numerous actual genomes, which would be the ones that carried only the orange star allele. The blue allele is very frequently present, but often in concert with other things, and certainly isn't the most numerous in terms of co-occurring with this orange star allele. So that kind of illustrates to you the concern that we had, that we're using consensus genomes, but we're missing underlying data. And newer long-read sequencing technologies like PacBio and Oxford nanopores, MinION, offer the possibility to get past this, but with the caveat that for now, they work best when you multiply sequence through a particular region in order to have full accuracy. And so I think for a big DNA virus or for large genomes where you want to know the DNA level, not just the transcript level, it's really important that we continue to consider hybrid approaches that would have long read scaffolding and short read for sensitivity and accuracy. But now I'll tell you what else we saw when we sequenced this population, which, if you remember, wasn't just about asking if siblings were in fact different, but asking how the sibling viruses might do if we let them compete with one another. And I told you that these syncytial ones kind of took over. And it could be that there's just one syncytial variant that's really fit and takes over the entire population. Or it could be that there's lots of distinct things that are competing with one another and coexisting, but that everybody by the end of the day acquires a syncytial mutation. And actually the latter is what turns out to be true. There's no single one thing that wins and has a selective sweep. It's a big mixture. So this is another depiction, cartoon, of course, of the viral genome and an identity plot up top, for which the only thing you need to take home is to say, if a bar is gray, it was nearly 100% identical across all the passages we sequenced. So a lot of the genome is pretty identical. But if we look for those minor variants or places where there was allelic variation at a particular locus, and we put a little tick mark on this horizontal line, anytime there's one of those allelic variants, and we sum that up across 10 passages, I hope you can see that there's lots of allelic variation underlying the overall consensus identity. So that allelic variation by the 10th passage is represented down here. There's a lot of different mixtures of alleles in that population. They're not all on a single genome. And we've published this work, so I'm going to just gloss over some of the other aspects of what we noticed in there. But I want to just show you as some examples that there are things in the population that are either selected against, like this blue line, a kinase mutation, and others that seem to be selected for, that are either contributing to enhanced fitness over time or are piggybacking along with some other selected allele. And it was interesting to see that while there are certain functions that we think might be favorable, either to lose or to gain in these sequential passages, it didn't seem to matter which way the virus got to its intended goal. Almost every virus in the population sooner or later developed a nonsense mutation in UL13 that broke the function of this kinase gene. Many of them acquired a non-synonymous mutation in another gene called UL14 that we don't know the impact of. And everyone acquired a mutation in glycoprotein B, which is from the gene UL27. And those, it turns out, are the ones that cause this syncytial phenotype. So this was really instructive for us to really see in a lab-based setting the way that competition plays out and the way that the virus might have fitness advantages for a particular outcome, but it's not tied to any particular genetic route by which it has to get there. It can be quite flexible. And so we've taken all of those things that we learned in a lab setting and are pushing them in the last few years to study as best we can this virus with the natural ecology of real humans, right? And we cannot run controlled experiments in humans. It's much more an observational natural ecology exploration, but I'll tell you a little bit about that data next, and then end with just 2 slides of insights about neuronal and animal models. And I think we're doing well on time so far. So a lot of the observations I told you about, I gave them to you in the context of the lab, but I didn't dream them up because I actually cared a lot about viruses in the lab. I'm an experimentalist, so I do love studying things in the lab, but I also study them because I want to know and understand things that happen out there in the real world. And in the real world, I told you that people who get this virus have all these different infection outcomes. So I'm going to talk to you a little bit about some of the things we saw when we started working closely with a clinician, Christine Johnston at the University of Washington. And the 2 figures I'm going to put on this slide are from her paper, not mine. So I want to give credit to her and her colleagues who did that work. But these are some data showing the penetrance or the severity of viral genital infections. And this study was using HSV2, but she has another unpublished set of work with HSV1 that looks much the same. So these illustrations are showing you about 30 days, I think it's actually 25 depicted here, almost a month's worth of viral shedding data for 2 individual women. Patient number 1 has almost no shedding. There's 1 day of what is called asymptomatic shedding. The person didn't even have a lesion, but swabbing of the genital area, for which this is a diagrammatic grid of her genital area, showed a little bit of viral shedding at a low level in a couple places. In contrast, patient 2 is an example of a person who's suffering a lot. In this approximately month-long period, she has multiple outbreaks of lesions, that's what the L indicates, other days of asymptomatic shedding, and when she's having an outbreak like these, she's shedding heavily in the sites where she can see a lesion, but also many other sites in the genital region, right? So this is a high opportunity for transmission to other individuals and highly unpleasant for the patient herself. And we wanted to know more about the natural history of this in the perspective of the virus and viral genetics. Is it always the same across time? Are there spatial differences in the shedding here up in a distant area from the actual lesion site here? Those are the kinds of questions we wanted to ask in this human setting. And part of the reason to ask that is that we want to understand why human symptoms differ so much from person to person. All of the individuals in this study led by Christine Johnston have fully normal immune responses. They are immunocompetent to the best that we can see. So her data, as well as other twin-based studies, have really suggested that viral features and probably virulence genes in those pathogens do influence the outcome in humans. But it's certainly true, this purple is what I study here in my lab, but it's certainly true that there are human genetic susceptibilities that have been shown in rare cases to influence extreme human susceptibility to infection, such as knockouts in TLR3 can lead to very unfortunate and early losses in terms of humans succumbing to infections that are ordinarily handled fine by others. And instances like chemotherapy or life stresses like a heart attack can also shift the likelihood of having an outbreak of a herpes virus or your susceptibility to a new one. And that same type of heterogeneity in terms of susceptibility to infection and infection progression has become very common in terms of national media reporting right now during this pandemic. And we see and know that not everyone suffers equally when they're infected by this current coronavirus. So as we went into studying humans, I want to give you kind of a big picture, and then I'll give you a couple tinier examples. At the big picture, we've had a number of papers over the years just trying to define how much heterogeneity is out there on the planet for these viruses. So I told you that these viruses can be 3 to 4% different genome-wide, and I showed you a phylogenetic tree with maybe 10 different viruses, of which it was probably 4 or 5 independent ones, and then some of those sibling groups. But we've expanded that drastically over multiple studies, and I'm showing you a figure from one particular one, but just to illustrate what it looks like when you've added up to 100 or so. And also to tell you that we've noticed, although it's fading with time, that there was a geographical bias in terms of the viruses that we were isolating from different parts of the world for HSV1, where viruses isolated in Europe tended to look more like other European viruses than ones gathered, let's say, in Kenya. Whereas for HSV2, there was no such pattern. It's a quite different species. It has crossed over into the human lineage much more recently from another primate ancestor. And there's no geographic bias that we can see in this virus. Also, these viruses have far less heterogeneity. So I'm not going to say too much about HSV2, but just wanted to give you that big take-home message. If I adjust this unrooted phylogenetic tree, which I should have explained, that's why it has this spiky appearance, is that it's an unrooted graph of the phylogenetic network. If I fix it so that they actually have the same scale, this one for HSV2 would actually look very tiny and hard to read. And that's because overall globally, HSV2 has far less genetic divergence. And that has to do with the fact described by work of others, Wertheim and colleagues in 2 other papers, that it's estimated to have crossed into the human population much more recently than HSV1. But I told you I wanted to study not just the existence of diversity between humans, but why one human is suffering more than another. And to do that, we had to bring in a few more technologies. So one of the most enabling ones, which I imagine some of you are using as well, is to use what's called in-solution enrichment, or oligonucleotide baits, that are biotinylated and designed to match the target genomes that we want. And so we can take HSV-specific baits, for instance, and mix them with someone's skin swab. And then through PCR and isolation on strictavidin-coated beads, you can pull out the biotinylated baits and the DNA they've bound to, and work with that enriched population for sequencing. And it's not perfect. We are not yet to the point where we can pull DNA off of someone's coffee cup or water glass and sequence a genome, but we're not actually far off from that. And so the lab has published a couple different papers now where we've used exemplar, extreme human case studies to kind of develop this technology and show that we could do this, that we could sequence from something as sparse and low copy number as a skin swab, as well as sequence from viruses in culture, which we've been doing for years. And so we described that case of female genital shedding, the really extreme example, in a paper a couple years ago, and then showed that we could also apply this to other kinds of extreme cases that are truly unusual. Like in this particular one that I'm depicting the cartoonized diagram from, it was a dual fatality, where there was a very unfortunate case where a mother progressed to a severe infection and was viremic at the time of delivery, and her neonate and her own infection went unrecognized for too long, and both of them lost their lives. And we were asked to just look at the virus and say if there was anything highly unusual about it. And the unusual things we saw exist, but it's hard to say that they're absolutely causative of what happened in their progression. But what we could also say was that there was diversity in the mother's infection, most of which was passed to the baby, but not all. And the baby, even in just 5 days of life, developed quite a few new variants in that highly naive and not fully immunologically developed environment. So it's really been a good way to open up the windows of possibility for studying this virus in humans. And I'll just give you 2 quick highlights of that because I want to leave ample time for questions. But if I continue to use that unrooted phylogenetic network as a way to just give you an overview of these viruses, I want to highlight that when we were doing that genital study of a particular patient with a very severe infection, we sequenced more than 10 viruses drawn from her swabs or from cultured virus from her body. And they all form a branch on the tree that is particular and different from every other virus. So that is perhaps not surprising. But I hope you can see that it's a fat branch. And these viruses are not all alike. So over time, she's shedding subtly different viruses that, just like those sibling populations I showed you earlier, perhaps they were on the order of 97 to 98% identical. But the 1 to 2% difference spread across the genome still left lots of room for coding variations to occur in her virus over time and potentially be transmitted to new individuals. And that point of transmission is the focus of ongoing studies in the lab. And we've been looking at both transmission cases and additional cases of new genital infection, in part to ask whether or not it was significant that this one initial first person we sequenced formed this new branch on the tree. We kind of wondered, would all human genital infections be in the same part of the tree? And the answer was no. Different new human genital infections span the whole pantheon of possible genetic diversity for this virus and create their own new branches, like these viruses that are different from anything seen before, but others like these that are very close to previously sequenced oral isolates and are just appearing now in genital niches. And I'm showing you single point sequencing from each of the humans on these red dotted branches. These are all new genital infections. But as we sequence deeply from each of these individuals and further unpublished work, they all form fat branches like this, where sequential isolates over multiple days or months drift slightly from the overall consensus within that patient. And that really echoes the same things that we had seen in lab, but are now showing you that that wasn't just a lab artifact, that happens in people too. So the last thing to say is that as a virologist, I needed to be able to demonstrate that what I was seeing in the lab was relevant to people and be able to understand it genetically, which is what genomics has been really good for. But then we have to be able to kind of circle back into the lab and say, okay, so now we observe that variation really exists in people, it's there. How are we supposed to interpret which bits of it matter to clinical outcomes? And we can infect people on purpose. So that's where we use neuronal models and animal models to come back into the lab and demonstrate that there are consequences to these genetic differences. So in some work that just came out this year, we took 3 of the same different lab strains that we've been studying early on. They're called COS, F, and McRae, but it doesn't particularly matter here. And showed that when we infected neurons, both viral and host gene expression varies per strain. And this is a plot of all the different viral genes or transcriptional units that we were looking at. And the only part to take home from this is that there's a lot of different colors on there. These viruses express their genes at a different pace and actually at a different level, depending on what strain it is. And we verified some of those at the protein level. So it's not just transcription. And in terms of the neuronal transcriptome, so the host response, those genes are even more numerous. So I can't fit them all on one slide, but I just wanted to say that if we do a typical PCA or principal components analysis for those neuronal host responses, they also segregate and differ between strains, as well as over time. And as another example, when we put these viruses into intact animal models, which is granted different than infecting a human, right? So a mouse does not have a co-evolved immune response to these pathogens. It nonetheless provides another important window on responses to infection in a way that cultured neurons in a dish don't have all the complexities of any immune response or any intercellular interactions with different cell types like astrocytes or microglia. So earlier, back in 2017, we had done one of our first transmission studies using cultured isolates from a father and son. And as part of that paper, we show that those father and son isolates are nearly identical. They have just a little bit like the other sibling viruses, although these came from different humans, they're father and son. They have just a little bit of genetic difference between them, but when we put them into animal models, they function nearly identically. They actually are highly attenuated by coincidence, as compared to a more virulent strain called McRae that is extremely lethal when introduced into mice. But what we've come back and done since then, and an unpublished work with my colleagues Nancy Sawtelle and Rick Thompson at Cincinnati, is to show that if we took more than just this one pair of isolates from the father and son and something highly virulent like McRae, but took a whole number of independent clinical isolates and examined them in this common host background, you wouldn't just see 2 outcomes. It's not binary. The real world has gradations. And so there are viruses that are extremely attenuated, but maybe not quite as attenuated as the father and son viruses. They are somewhat lethal in mice. Other viruses that are extremely lethal and ones that are in the middle range. And we're using genome-wide association techniques and further studies to try to isolate the viral genetic loci that lead to those differences. So I think it's important to say that, and of course I should mention here, these animals are genetically identical. So they provide a way to be sure in a way that you can't in humans, that there wasn't a host genetic component to what you were seeing in terms of human to human differences. But I just want to mention here that it's really important to be able to take all the genetic diversity that we see and bring it back into these lab-based models in order to see and test these outcomes. I really think that all of the different techniques that we use in the lab don't function well in isolation for us. We get the insights we get because we're using all of these different approaches. And so I would encourage you to be similarly encompassing in your own work to think about how you can use different methods to broaden the interpretations that you can make from your data. So I think that the key questions are ones I've mentioned already, but I just like to put them there in writing if I'm talking too fast at times to get them across, that we're very interested in figuring out how human and genetic variations lead to the whole spectrum of disease. And part of that is because we'd like to be able to predict the clinical course of disease, right? If you have a newly infected adult, or even more critically, a newly infected baby, then you have the ability to say the virus they are carrying has been observed to be highly virulent in other individuals. or in contrast, highly attenuated. Perhaps that would allow you to debate the degree to which you might try to treat that neonatal infection, or whether you would advise that adult to use, let's say, daily suppressive therapy to hold back their infection, or advise them that their infection is likely to taper off in symptoms in a year, and if they can handle it, they can probably manage their infection in that way. And we would, of course, like to be able to provide and inform the development of new diagnostics and therapeutics. If in the course of what we're doing, we discover other highly attenuated viruses, that may lead to useful outcomes in terms of live attenuated virus vaccines. Similarly, genetic sequencing might reveal parts of the viral genome that are incapable of adaptation. And if we can target those with an antigen for vaccination, perhaps that's an area where the virus can't evolve to escape a newly developed vaccine. And so I just want to end by thanking again the people in the lab, because I just gave an overview of many years of work. So these are the folks currently in the lab, and the two I have pasted up here on the right are two recent graduate students whose work is nonetheless heavily represented in what I talked about, since our lab has really only been at Penn State for about 7 years, and our various collaborators. So thanks, and I would be quite happy to hear questions. Would you like me to leave the slides up, John or Gus, or shall I take them down while we're doing questions?
You can leave them on while you answer, in case there was some specific comment you could make, Maria. Yeah.
Okay, sure.
And so Andrew Lewin, you sent that directly to me in chat. Do you want to go ahead and just unmute and ask her the question directly?
Hi there. Thank you so much for an awesome talk. I really enjoyed that.
Oh, good.
I had a few questions. I don't have to ask them all, but it kind of depends on how long the answer is, I guess. The first one is, so as you know, HSV has a relatively high number of known variants compared to related viruses like canine herpes virus and feline herpes virus.
Yes.
What do you think the best explanation for this is?
Oh, you've asked a really perceptive question. So I don't know if you work with those other viruses, but I should say for the sake of others, that Andrew's asking about comparisons to the quantity of diversity for HSV1 versus for some of these animal herpes viruses, like I think you mentioned canine and feline. All of our pets have their own herpes viruses, and actually all of our agricultural animals like bovine and equine, actually some of them have multiple. And Andrew's asking what I think about the fact that HSV is even more genetically diverse than the others. And honestly, Andrew, my answer would depend on the day, because sometimes I think, oh, we're just unlucky. Like, this is just a mess. You know, we humans have diverged so much from our ancestral evolution in, you know, presumably somewhere in the African continent, and we've diverged, and we just diverged the viruses with us. But other times I'm willing to leave out there the possibility that what we know about canine and feline herpes virus, for instance, think about what they are. They're pet herpes viruses from domesticated animals that humans presumably domesticated in relatively recent times. And I kind of wonder if you had a globally distributed wild animal species that was actually as successful as us at moving around, that we could sample as well as we've sampled ourselves, we humans. Maybe there are animal herpes viruses that are just as diverse. It's just that they're understudied, and relatively speaking, they haven't had the ability to be both globally distributed and then to move, you know, numbers of those animals. Like, let's imagine wolves. You know, they're not moving from continent to continent in the way that we are. So I sort of feel like it's hard to disentangle the bias of observation from, you know, the very unique status of humans on the planet, where we both move the most, do the most, change the most, and study ourselves the most relative to any other species. So that's my guess. Does that kind of answer your question?
Yeah, I think that's, yeah, I totally understand why it's a guess, and I think it's a pretty good explanation. The other thing, I just had one more question, I guess, that you maybe could answer for me. I thought it was really interesting when you were talking about the drift that you get in culture of the certain HSV isolates, and how you think that there's variants that kind of drive that. Is there any kind of evidence for that kind of drift in culture for other herpes viruses other than HSV?
Yeah, surprisingly there is. So one of the blood-borne herpes viruses, human cytomegalovirus, actually made many observations about this exact same thing years, if not a decade, before the human herpes simplex virus field caught up. And that's because a number of years ago, Felicia Godwin and other colleagues in the, I'll call it HCMV because it's shorter to say than human cytomegalovirus. She and others in the HCMV field realized that clinical isolates found in humans had a chunk of the genome that was missing from most lab-grown strains. And then kind of checking out the history of how did that happen? And what was that chunk for? Work from her group and many, many others have figured out that a fairly large chunk of the HCMV genome is lost because it creates differences in terms of the entry receptor that are highly favorable in humans for the naturally infected cell type, but not useful for the typical cell type used to propagate that virus in vitro. So they totally set the playing field for things that I would say are much more subtle in HSV1. We have not seen any signs that there's 10 kb of the genome in human infections that are missing for when we grow HSV1 in a dish. Although, back when I started sequencing, that was something we talked about, is would we find the same thing? We just didn't know. But it's interesting and important to keep an open mind, because what they found in terms of that loss wouldn't have been found except for very careful biology and carefully keeping track of what they saw as they move between different cellular models. So I try to really encourage trainees to keep a very open mind and not throw away data when it doesn't fit their pattern, because I think these viruses have lots to teach us. And if we're not willing to keep an open mind, I feel like we're going to continue to miss things.
There's a question from Michelle. Michelle, you want to ask your question?
Yeah, sure. You mentioned that there was pretty high GC content in these viruses. I'm just wondering if you've looked at methylation at all as a regulator of different strains of virulence.
Yeah, so I have wondered about that over the years, and we have not circled back with newer techniques. So I've had a couple students or postdocs in the lab say, yeah, I'm curious to do that, and then not actually be able to kind of finish and get that done. But it had been looked at before, and it has not been noticed to be a regulatory mechanism. But I think, again, it's quite possible, knowing what we know now about the genetic diversity that can be in these viral populations, that if we weren't doing it carefully or with a very homogeneous population, we might have missed some of the signals that were there. So I totally feel like it's something that we should come back to as we move forward. And I'm gonna I'll leave the slide there, but I'm gonna move this around because it's weird how I lose my mouse when we're in full screen mode there.
There's a question also by Claire. Claire, you're gonna ask your question?
Sure, I'll ask my question. This is kind of a self-serving question, but do you have any tips on how to better sequence over these really diverse regions of the HSV1 genome?
I have, plenty of tips I could say from the trenches, right? To say, here's all the things we have fought through and tried. But I think it really depends on what your sample is that you want to study. So, for instance, we now started using these oligonucleotide enrichment baits that we use to sequence sparse samples from humans. And we didn't used to use them for cultured viruses because we didn't need them. When we grew viruses in culture, we could get lots of DNA. But since we designed them and had them in the lab and used them for human samples, you buy a set of baits and then you can dilute them into many reactions so that they kind of cost a bunch when you buy them, but you can really stretch them out in a long way. We realized, oh, well, we could use that on our cultured viruses also. And that lets us sequence genomes from a much smaller volume of cultured virus than what we used to grow. So it used to be that when we wanted to sequence a cultured virus, we would do an infection and grow up like almost, micrograms of DNA, 0.5 milligram. For those of you who work in a lab, that's a lot, especially from a virus. But now we can use just a little tiny aliquot of stock that is nowhere near that amount, nanograms of DNA from the virus, and still get a genome. And so I think that helps because you can sequence your cultured virus in such a way that you don't have to passage it as much and let those issues develop. And in terms of the repetitive parts of the genome, I have to say I'm still pushing hard to make that better. But I really have high hopes for combining Illumina and nanopore technologies. And that's not going to be super easy to say everyone should have a nanopore and Illumina in their lab. This is super accessible. It's not, and it's really being done the best by Pete Maas's group over in Belgium with HCMV. They continue to push the envelope faster than HSV does. And I was hoping to go there for sabbatical to kind of figure out some methods and build a new pipeline. And that got postponed, of course, by the pandemic. But they continue to do their great work, and I hope to learn from them next year. But I think in the meantime, Illumina still provides the best way for most parts of the genome, but for one of the genes that, Claire, I think you care about most, that's in one of the worst and hardest parts of the genome. And so I don't have a specific Band-Aid for your specific part of the genome. I can just tell you that we have had such trouble with the region of the genome that Claire's going to talk to you about in her next talk, that we often go after it with straight out PCRs and even old fashioned restriction fragment length polymorphisms, or RFLPs, the same things they use in DNA forensics for humans. And it's imprecise, but a good estimator of length differences at those tandem repeats in that region. So if there's something that you know differs between strains and you want to figure it out, there are tools like that that are imprecise but highly informative. And we could show you the things that we haven't published but have learned in that region, because they're sitting on a shelf because they don't quite fit in a paper sometimes, but might be quite useful to you or to others.
And the bigger issue is, I guess, the number of inverted repeats and the fact that both L and S components invert. So it becomes quite difficult thing to see which of these prototypic arrangements you're actually sequencing, because there are 4 different variants. And that becomes very complicated. I could ask a question, Moriah, to fantastic work, and I think you're really in the leading position to understand those really highly difficult genomes. You mentioned the syncytial phenotype, and I remember from textbook references and also from pathology slides that if you take a look at a lesion in a particular sore, what you actually see is fused cells, syncytia, even in wild type viruses. And of course, the misunderstanding sometimes in the reviewers of grants to say, well, this virus you isolated is now wild type, so the syncytial is really an aberration. But in fact, all herpes viruses, when they're really in their pathology themselves, are actually syncytial. So the question is the syncytial phenotype associated with virulence? And that's been up and down depending who you ask. So what do you think about that?
Well, for fun, let's put those nice pictures back there so at least people can see them. I can actually replay them if anybody wants to see them again as they're on here. Oh, I guess they're only going to play one at a time. Well, in any event, my thought about syncytia is I'm glad to hear that you're one of the people who has an open mind about syncytia being useful, because I agree. There's a bias in our field that says they're not good for anything, or only varicella zoster virus, which is that one, remember, that I said causes chicken pox and shingles, and the pox can look like fluid-filled lesions, and so that's can create this sense that, well, syncytia matter for that virus, but not for HSV. But there's actually really cool older data that had demonstrated years ago by putting viruses in mice. Gus is nodding because he knows this, but I'll say it out loud for the benefit of the others, that if you take syncytial viruses like the one on the right that are only syncytial, and ones that are like the one on the left and only CPE inducing, and you put them into equivalent mice, the one on the left can infect and cause fatality in that mouse, whether you introduce it at, let's say, the foot pad or the flank or the eye, or if you inject it right into the brain, which is unnatural, but fine. Whereas the one on the right, the syncytial one, can't cause disease if you put it on the foot pad or the flank. It's like it can't get past the immune system. But if you do the unnatural thing of injecting this pure population into the brain, it's super lethal, and super lethal with not too much virus. And so I trained as a neurobiologist before being a virologist, and did genomics the whole time in both of those places. So I have always been intrigued by this finding, and have long thought that it would be a really smart thing for the virus to keep a mixed population, like the population that gave birth to this mixture, and have just a few syncytial viruses around so that they can both get past the peripheral immune system, and when they get to the brain, do as many things as they want in the brain, if that's the best end goal for the virus. And of course, when we put these viruses in mice, it's an acceptable model, but mice don't transmit to one another. It's a zoonotic infection, and most of the time it will kill them, right? So we're using these mice as a model for what might happen in people. But in people, we have isolated, in a couple rare instances, syncytial viruses from humans. And the only times, both of them were likely AIDS patients, although it wasn't recognized at the time. But both times they came out of humans, those humans were carrying a second strain that had this kind of cytopathic effect. So it at least sets the possibility that, yep, they could coexist in humans. They would probably also be in the minority, and they would coexist with something that isn't syncytial. And if we just look carefully, we might see them. So I'm with you, Gus, and I don't know, aside from what I just told you about, maybe they're helping the virus in certain situations. I feel like we're still digging into that. But if you're curious, Stefan Oliver and another scientist are running a special issue at the journal Viruses on syncytia and about their potential actual biological and pathological inputs and what they're good for. So that's a place whether you want to submit to it or whether you want to read the issue when it comes out with multiple papers that will, I hope, reflect on that some more.
Yeah, I wanted to also, obviously you have a two-body problem, right? So you would expect that immune status would actually affect the evolution of those viruses, and the expectation will be that perhaps even regulatory genes that modulate the immune response maybe have a faster rate of evolution based on immune reaction. For instance, type 1 interferon response, a number of different genes to the virus. Do you see a different rate of evolution, more human isolates on those immune regulatory genes, or is it across the board? What type of classes of genes, whether it would be, regulatory or others actually affected? Are they affected in the same rate, or are there differences?
Well, so I would say I'll use this as a quick image to point to that it has been really easy to see when we compare different isolates that there is the most amount of variation in the surface proteins of the virus. So this is perhaps not surprising. And I know your lab is one of the ones that has contributed to knowledge in this area, glycoprotein K, glycoprotein H, and many others. And that's not surprising to say they're some of the most different from person to person because they're going to be the ones seen most easily by the immune system. But if I was following your question right, I think you were also kind of asking about a different tier of viral genes, which might be the viral genes that specifically modulate the host immune response. And some of those are in those particularly hard to sequence genetic regions. I don't know if I can easily find it, but in any event, the most repetitive parts of the genome, there are these structural repeats that contain 3 viral genes that exist in 2 copies each. And those are some of the key regulators for turning on and off the virus and for helping to modulate entry into latency and reactivation. And those genes are actually the least well sequenced just because they're extremely GC rich and highly repetitive. So it's both one of the most important places where we would expect to see variation and one of the places where the window we have onto the genome is the most cloudy. And we have yet to kind of smooth it off and get access to those because getting full access to those genes often requires those extra techniques I was telling Claire and you about. So I think for sure surface variation, but also more to come in terms of the genes that might co-evolve with interferon control, trying to combat interferon output from the humans. But I feel like we should probably let Claire get on to her talk.
Yeah,
and
absolutely.
I'm happy to answer other questions later.
Yeah,
too.
thank you, Moriah. And there'll be additional questions. We will, I'll try and answer them later. So, Ramesh and Chris, we can move on.